in this case taken from lymphoblastic leukemia in children, some resistant to other treatments, creating a culture system ad hoc suited to this particular type of cell. It was seen that in most cases there was sensitivity to concentrations achievable in vivo in patients. So there may well be non-cytotoxic mechanisms involved. We're not yet sure about the administration of other compounds together. Previously we were talking about plant extracts and such. We don't know to what extent those substances, extracts may interfere. For many years I've been involved quite successfully as well in this drug that's now been registered in Europe for sarcoma, soft tissue sarcoma, and ovarian cancer. Again, it's a natural product. It's highly complex in terms of its mechanisms of action. Besides the cytotoxic effect, it also has effects on certain cell populations, normal cell populations, which probably are very relevant due to the anti-tumor effect. This is the tunicate that I was talking about. It's a marine compound. Someone this morning was saying that there was much research into substances capable of protecting against sunshine. These tunicates initially were studied by the American Army to try and find substances to protect against radiation. Many substances were identified, including this one, whose structure is very complex, and studies were conducted up to the early clinical experimentations with an extract, after which a Nobel Prize winner from Harvard purified the product, and now it's obtained by a synthetic process. It binds to DNA rather differently to the drugs that we generally use in anti-tumor treatment, because it doesn't bind to the main groove, but to the minor groove with alkylation. It's not the site where most alkylating agents bind with. And it's interesting to note that based on studies by Lawrence Harley and his group, it was also found that a part of the molecule protrudes outside of the DNA, the minor groove, which led us and others to think that the mechanism may be even more complex. It's not just a damage caused to the DNA, but an injury to the DNA, but also involving the proteins related to DNA that don't fail to recognize the sequencing, the regulating sequence. This can be found in vitro. There are a number of genes where the transcription factors that should bind to certain sequences with sense fail to recognize the sequence, and so there is an abnormal transcription regulation. This is the another study, and you can see that it's possible to switch the transcription, taking it to 37 to 42 degrees C in the presence of nanomolar concentrations of compound that block the process. So these studies lead us to believe that the drug might have different actions to others already seen, and one might ask about the selectivity of this drug. In reality, the number of genes altered by transcription was different, but then it was seen that this effect did not relate too much to proliferation. Some cells proliferated very quickly and others proliferated slowly, and there was no clear relationship Typically, monocytes and macrophages, these cells do not proliferate, but in these cells, our group and Mantovani's and others found that it was a strong modulator of cytokine and chemokine production. And this was interesting because there's a line of research going on inflammation and cancer and the role of cytokines and chemokines and anti-angiogenic factors, etc., that can be produced in tumors. And this is the tumor-associated macrophages, and they're very sensitive to the drug. And here you can see the secretion of CCL2, IL-6, VGF, and ANG2, which is strongly downregulated in this situation. So then the drug went on. It has a strong anti-tumor effect, both on sarcomas and ovarian cancer. And this was an experiment conducted at our lab, and you can see that the drug produced quite a significant remission in the xenograft at lower doses. It was active, and so it went then on to toxicology. The problem was that in toxicology, it emerged that there was a problem of liver toxicity. Here we looked at the mechanisms, and again, we were dealing with inflammatory mechanisms, and Professor Gescher has been mentioned quite a number of times. At the time, we were working with him in a hepatotoxicology unit, and we found that the earliest events, this was in rats, were related to damage to the biliary duct, which then turned into overt liver damage. And the liver, the gene expression changed in the liver, and this was a system whereby we were able to study in the rat what was going on. It was found that liver toxicity was quite important, and therefore we needed to find a way to reduce it.